All right, guys, today we are going to look at some material from your fourth unit. So that can be good news or bad news, depends on how you want to look at it. It means you're going to be starting your fourth unit, which is your last unit, which means you're close to the end. So that could be good, or if you're not prepping, it could be bad, you know. So fourth unit will be material that will cover right up until the end of the semester, and then we'll go with a Unit 4 exam, and then we'll have a final right after that. So it's being bang boom right there at the end. So next week your Unit 3 exam is on Thursday, because that's your off non-lab day. So we'll be taking that, your Unit 3, excuse me, exam next week on Thursday. And we will be looking at a little bit more of this information. I'd like to get my projector working. A little bit more of this Unit 4 information when we do. All right, let's see here. This shouldn't be too much of a problem for you guys, because it's really kind of building on what you've seen with the cardiovascular system. We were looking at the heart as a pump, and what we're going to see today is the vascular structures in which the blood moves through. One of the things that we looked at the other day in lab was we talked about arteries and veins. As this ring a bell, we said arteries are structures that take blood away from the heart, veins are structures that return blood to the heart. And for the most part, you have this association of arteries having oxygenated blood in them and veins having deoxygenated blood in them. But remember, with the heart, that's a little different. We have some structures that were veins that were oxygenated and arteries that were deoxygenated. So today we're going to look at the characteristics of arteries, veins, and to a degree capillaries. And it's not going to be all that difficult for you. You want to appreciate that your arteries, again, carry structures away so they're under pressure, and that your veins carry blood back to the heart, and you're going to find they're not under a great deal of pressure. And for that reason, structurally, they're a little bit different from one another. Your capillaries are very, very tiny. This is where we do gas exchange. So last section I was working with today, we were talking about capillaries, and students were having a really difficult time trying to envision just how much we have in the way of capillaries. But our body is covered with capillaries. So when you think about the size of them, they're microscopic, as are most of the structures that we're looking at. They're going to be comprised of simple squamous epithelium, thin cells, one layer, and they're going to be tiny in diameter. The lumen, the opening, is very, very small. And so if we were to take all the capillary beds in your body and look at them, if we could actually visualize them, you would see this outline of your physical form made up by these capillaries. But remember that you only have about 5 liters of blood moving in your body at any given point in time. And even though 5 liters of blood sounds like a lot, if you try to push that blood through all of the hollowed spaces of the arteries, veins, and capillaries that you have, you don't have enough. There's not enough blood by volume to fill all of those tiny little capillary beds in your body. So we're going to show you how that works here in just a few minutes when we get to small sphincteral muscles around them. When we look at these vessels, you're going to hear that they're going to be comprised of 3 layers, 3 tunics. Do you guys remember the eye, the 3 tunics of the eye? Well, you're going to have 3 tunics here. You're going to have an external tunic, you're going to have an internal tunic, and you're going to have a middle tunic. So that's what you're seeing right here is a description of these different tunics. The intima is the innermost, the media is the middle layer, if you will, and the externa is the outermost layer or tunic to your vessels. So let me show you what's going to be important. This is kind of where I would be making note of what I need to know about arteries and veins. When I look at the 3 tunics of these 2 different vascular structures, they're going to be very different. So here are your arteries. They're going to be comprised of this outer thin tunic. That's what you see right here, the tunica externa. Then we're going to have this massive middle tunic, the tunica media, and that's where you're going to find an awful lot of smooth muscle, a lot of it. And then finally, the innermost tunic, the thin one, the intima, is also going to be comprised of a little bit of elastic connective tissue and then a thin layer of epithelial tissue. Now remember, the term lumen means the hollow space that is where the blood moves inside these vessels. So do you see how thin these outer and inner layers are, but how big and deep the media is in an artery? There's a lot of muscle there. Now let's go over here and look at a vein. On the flip side, the vein has a thicker layer. The externa is much larger. It has an incredibly small tunica media, very little muscle in there at all. And then your tunica externa is much thicker here than it is in your artery. Okay, so let's kind of understand why this is the way it is. When we look at your arteries, that's where the blood from your left ventricle is going to be found. So every time your heart ejects that blood, it's under a great deal of pressure, and it's pushing very hard on the internal wall of your arteries. So you have thick muscle in that media to keep the artery from expanding too much under that pressure. It holds, it's fairly strong. On the flip side, your blood goes from arteries, so they have, lumen like this. Now, obviously it's not this big, but you get the concept. You go from a pipe like this, and then you got your arteries getting smaller and smaller and smaller, and they move down to what are called arterioles. So the hollow opening is smaller. Then you get to the capillary beds where infinitesimally small, lumen, tiny, tiny openings. And then we gas perfuse there. Well, then the blood starts to come back to your heart. When it comes back from the capillaries, it goes into venules, which have a small lumen, to veins, which have larger lumen. Well, your blood is not under pressure there. You can't go from a big hose to a small hose and then open the hose up again and expect there to be much pressure. There's very little. What happens is your veins are going to be surrounded by skeletal muscle. So every time you move, that skeletal muscle contracts and the veins are kind of sandwiched in there, so that the muscle, when it contracts, pushes the blood and slowly but steadily pushes it back to your heart. That's why if you look at people that are sedentary or people that don't do much moving, what happens? That fluid pools where? In their extremities by virtue of gravity. That's why we want you to move, so you can kind of have your circulatory system assisted as far as returning blood back to the heart. All right, so how are we doing here with an understanding so far of the differences in the three layers? So you would want to know, if a question was asked you, what the tunica media meant. Well, that's the middle layer. And so you might have a test question that says, how is the tunica media of an artery different from the tunica media of a vein? And you would want to recognize the difference between these two. Here's the next thing that you're going to find kind of interesting when you look at your veins. They're going to have valves like this. So the blood is slowly moving in this direction. And as it does so, every time pressure drops, it kind of trickles here. But those veins, valves, catch the blood so that it doesn't go back and pool. Now, you'll notice that the veins, because they don't have that thick tunica media, they're not super strong because there's not a lot of pressure there. But pressure can build up there over time. And you see this sometimes as people get older, their veins get weaker and they stretch out a little bit more than they should, and then the veins, valves don't catch each other and close. How many of you are familiar with varicose veins? There's your byproduct of that.
Very thick veins.
Well, varicose veins are going to be where the veins themselves have stretched out too much and the vein valves can't close against each other. And so you'll have sections where you have a lot of blood pooled in the vein, and then they kind of expand outward and become more visible. All right? Questions? All right, yes?
What's it mean when your leg falls asleep?
Okay, typically what's happening there is you've got pressure on a nerve group that is causing if you will, diminished action potential from the nerves in those areas. And it just so happens that your nerves and some of these vascular structures are going to be close to each other. So you sometimes also have blood pool because of that compression. You've seen this probably when you hang your arm over the side of something, and then you'll notice over time the veins, which are closer to the surface of your skin, become more prominent. They appear to be larger. And that's because the blood is pooling in the veins, pushing them out in that area. But the loss of sensation is a byproduct of the compression on the nerves. All right, any other questions? So let's take this a little further. So we've been talking about these arteries and these veins, looking at the differences in those layers, the tunica, external media, and internal. Well, this tells you a little bit about what we just went over here. And this shows you in a cross section what you would expect to see. So here's your artery, big, thick tunica media. Here's your vein, very small. Now here's the next thing that kind of happens. Keep this in mind. Your arteries and your veins tend to parallel each other. So you have deep veins and deep arteries. But a lot of your veins in return pathways will start to spread out, and then they'll get a little further out towards the musculature. So, keeping that in mind, if I'm after, let's say, an artery like your brachial artery, here it's deeper, but your brachial vein is closer to the surface. So as you look at this illustration, deeper arteries, more superficial or closer to the surface are the veins, because they're trying to get close to those muscle groups which push on them. Now, we're going to be dissecting a fetal pig here before too long, and when you do, the pigs that we're going to have are going to be injected with a latex dye in their vascular system. Well, now I want you to think about this for a minute. Students typically, when they first see these, they'll be looking for an artery, and the artery will appear very small, but the vein that'll be next to it will be huge, very large. And they're like, why are the veins so big and the arteries? Well, what happens is this. When we inject that fetal pig's vascular system with that latex dye, we are under a lot of pressure, and it pushes on the arteries. But what do we know about their walls? They have a lot of muscle there, so the wall doesn't expand. But when we inject the latex dye for the veins, what do we know about the media? Well, the wall's very thin, and so the pressure alone pushes that latex out and through. So it appears to have a larger diameter than the artery does. Okay? Then we get to the capillaries. We said the capillaries are where we're going to do gas exchange. So now remember, the capillaries are going to be very, very, very small. Tiny little lumens, simple epithelium surrounding them. So there's not much to these guys structurally. But I want you to look at this arrangement here. We're going to come to the capillaries again in a minute and talk about their diameter. You know, here's that artery group, all right, so big tunica media, and then look at this capillary group. That's just basically an outer membrane and smooth, simple epithelium on the inside. It doesn't have those three layers. Because why? Well, what am I doing here? Gas exchange. So I want to be able to diffuse the gases across that surface area. So I want as little as possible blocking that gas diffusion. But remember again, these tiny little lumen, they're very, very small. So it's not as if I have a parallel network of capillaries. I have a network of capillaries that spreads out all over the place. Whereas the artery may come in as a singular structure, capillary bed will explode. There'll be lots and lots of vascular pathways. Well, here's this pathway of arteries that we were talking about previously, from the heart down to this capillary bed. So when we start, you have what are called elastic arteries. Well, what's that all about? Well, if we get to the tunica externa out here, and we start working our way in, we're going to find that by the time we get to the internal areas, there's going to be a decent amount of elastic, remember that connective tissue? Elastic connective tissue embedded in there in that medium. Why? Well, what's it going to do? Every time your heart beats and pushes that blood out, where's the pressure greatest? Where the pipe is big? Right here at the beginning? Yes, it is. I know, that's kind of confusing. You're thinking because the pipe's getting smaller. But what's happening to the vascular structures? I may come out with one, but now I'm going to branch off into many. So as they branch off, even though the pipe gets smaller, the pressure is going to start to go down. So the actual area where the pressure is highest kind of surprises you. It's where the pipe is biggest, but it's where it's closest to the heart itself.
So whenever you're creating those different branches, you're decreasing the pressure by increasing the volume?
Yes, taking off more of the volume.
Yeah.
So this is important, so kind of stay with me. So where do I want these elastic arteries at? Well, I don't think I want them right there at the aorta, do I? Where the pipe is big, because I'd be worried they'd come out and rupture. But here's where we actually find them. These elastic arteries, these guys right here, are here. You see where I'm pointing? That's the coronary arteries. And you're going, well, that's right off the heart. Yes, it is. But do you guys remember when we were describing this to you? How is it the coronary arteries fill? We said when the blood is ejected up the aorta and out to the body, coronary arteries are down here low, right at the base of the aorta. Blood's not going there, not under force. What happened? It's the negative pressure. So when the heart relaxes and the ventricle opens, the blood wants to come back in. The aortic semilunar valves keep it from doing that, and now the blood goes in to the elastic coronary arteries, and they can handle that pressure. Everybody okay with this? Okay. So then as we start moving further away in the aorta, guess what we find? more of the muscular arteries. Now, that makes sense because here the pressure is high, right? And we measure this. We talk about this all the time. These guys can't expand out too much or they start to rupture or bubble out. So we find these muscular arteries in the areas where the pressure is initially high off the aorta. And then as we start moving away, look what happens to the arteries. They get smaller. These are called arterioles. Now the pressure is starting to go down a little bit, right? Okay. Finally, we transition over to the capillaries. We do gas exchange. We bring the blood back through what are called venules. That's the small version of the vein. And eventually to the large veins themselves. Remember, they've got the valves. All right, we see the valves here. But they have a thin tunica media. All right, now, we're describing the story of blood movement to you for a reason. Recognizing that there are different kinds of arteries. Elastic arteries, muscular arteries, and then the arteries get smaller and go into arterioles. So why are we describing all of this to you? Well, let's take a look at some of those illustrations. There's the tunica media of an elastic. There's the tunica media, a lot of smooth muscle in the muscular. There we see that the arterioles are getting small and the layer of muscles getting even smaller. Here's why we're telling you about this, because clinically, this is what you're going to experience when it comes to understanding these vessels. These vessels, as blood goes out from the heart, they're going to be loaded with material, not just gases. And so as we start looking at some of the material that can be in the vessel, we find that lipid-based materials can be thick enough in there that as the blood slows down, that material can tumble and settle on the floors of your vascular structures. What we're describing here is a byproduct of a condition known as hypercholesterolemia. If our lipids, certain types of cholesterol, are wrong, off, high, then that stuff will settle and we will develop what's called atherosclerosis. You've heard of those plaques in the arteries, right? Well, that starts to block the movement of the blood. And that's when we get in trouble with oxygen deprivation to some of our tissues. So we have to bypass these blocks at times or open them up. And so angioplasty is the process by which we open them, balloon them, put these tiny little tubes in, inflate the artery so its diameter is larger and bypass that block. Or we have to bypass the block entirely by grafting vascular structures that go around these occlusions. Now, the third one that you see here is scary as all get out. This is what happens when the arteries, the tunica media, doesn't develop a consistent media later. Let me explain. You guys know what it's like to blow up a balloon, right? All right, so when we blow the balloon up, it expands evenly in all directions, right? Well, as pressure is going through our arteries, some of us, unfortunately, have a genetic predisposition where the media in the arteries is weaker in certain areas. And so as the blood pushes through there under pressure, the weaker areas, like a balloon, bulge out or bubble out. And that's an aneurysm. Those areas are then notoriously weak. And if pressure is not controlled, they can rupture. And if they rupture, you're done. You hemorrhage, you bleed out, and it's over. All right, how are we doing with this? So understanding our arteries and our veins and the differences between the walls is kind of important. And we come to that conclusion. How many layers do we have to the arteries and the veins? Okay, we have a tunica that's inside, we call that intima, the one in the middle media, and the one that's external. And we find the muscle typically in the media layer more often than not, and the difference between the veins and the arteries is the arteries have a thick muscular media and the veins don't. But the veins have something in them in the lumen that arteries don't. We call those valves to keep the blood from going back. All right.
Wait, which one has valves?
Veins. So here you go. This is what we were mentioning a minute ago. We told you the capillaries branch out all over the place. So take a look at the way this works. Here comes your tiny arteriole. And let's say this is an area of your brain that needs an awful lot of oxygen. So as we go to the arteriole, we branch into these little structures, and here are the capillaries, and they are all over the place. And so the blood trickles into these spaces, and we do gas exchange, and we go from oxygenated blood to deoxygenated blood, and we go back to the veins and slowly back to the heart. But what did we tell you previously? You have such a large vascular highway, you don't have enough blood to fill all of these capillary beds at one time. So here's what you do. You have these little sphincteral muscles at the base where these arterioles come in. And so, let's say you're out exercising and you're running a 5K. Your demand for oxygen